Dr. Michael Snyder opened the discussion by emphasizing the complexity of glucose regulation and how individuals respond differently to the same foods. Contrary to the common assumption that blood glucose spikes are uniformly harmful, he explained that short, transient spikes, like those after eating grapes or during exercise, are typically normal and healthy. A healthy glucose excursion involves a rise in blood sugar followed by a return to baseline within 30 to 60 minutes. Prolonged or excessive glucose spikes, however, especially common in diabetes, are detrimental and linked to cardiovascular disease and other health complications. This time in range metric, where glucose ideally stays between 70 to 140 milligrams deliters, serves as a practical guideline, though individual variability remains significant. Further, Snyder illuminated the surprising variability found among individuals when consuming different carbohydrates. For instance, some people spike insulin in response to potatoes, but not grapes, potato spikers, while others show the opposite pattern. This discovery challenges blanket dietary recommendations and underscores the importance of personalized nutrition. Continuous glucose monitors, CGMs, now widely accessible, enable people to monitor their unique glucose responses in real time, revealing personal food sensitivities that cannot be accurately predicted by traditional measures like glycemic index charts. Such personalized approaches can help individuals optimize their diet and metabolic health, rather than rely on generalized advice about good or bad carbohydrates. Exercise's role in managing glucose and metabolic health was a prominent theme in the conversation. Snyder's research showed that exercise timing might need to be personalized based on a person's metabolic subtype. While traditional wisdom suggests afternoon exercise may optimize performance, his data indicate that those with muscle insulin resistance might benefit more from morning physical activity to improve glucose regulation. The form of exercise, whether resistance training or aerobic, is also an area of ongoing investigation, but Snyder highlighted the well-established notion that any exercise is better than none. He stressed the importance of exercise snacks, short bursts of activity, or muscle contractions throughout the day, especially for people with sedentary lifestyles. Even low-level muscle engagement, such as seated calf raises, can significantly improve glucose uptake. Taking post-meal brisk walks was emphasized as a simple and effective strategy to blunt glucose spikes. Walking facilitates glucose uptake into muscles, thus reducing prolonged hyperglycemia. This integration of exercise, both scheduled workouts and incidental movement, plays a critical role in metabolic control and overall health. A groundbreaking segment of the discussion was dedicated to the heterogeneity of type 2 diabetes, which Snyder subdivides into distinct phenotypes based on underlying mechanisms such as muscle insulin resistance, beta cell defects, insulin production or release defects, and incretin hormone (GLP-1) dysfunction. These subtypes have important implications for both lifestyle interventions and pharmacological therapies. For example, Snyder, a type 2 diabetic himself with a beta cell defect, found that building muscle mass through resistance training did not improve his glucose control because his issue lies in insulin secretion rather than sensitivity. The ability to categorize individuals by their glucose regulation subtype using simple glucose curve analyses from CGMs promises to revolutionize diabetes management. This precision medicine approach helps tailor drug prescriptions such as GLP-1 receptor agonists or other insulin secretagogues and optimize behavioral modifications. Such nuanced understanding also explains why some people who are thin can still develop diabetes, and conversely, obese individuals can maintain good glucose control. These findings challenge the simplistic association of body mass with diabetes risk and emphasize the complexity of metabolic health. GLP-1 receptor agonists, drugs like semaglutide, Ozempic, and its analogs, stirred a passionate conversation. These drugs have proven highly effective in improving glucose control in type 2 diabetes and often induce weight loss by suppressing appetite. Snyder shared his personal journey with these medications, reporting dramatic improvements in hemoglobin A1C levels and significant fat loss while maintaining muscle mass through frequent resistance training. However, side effects like nausea and concerns about excessive weight loss prompted him to moderate his usage. There's growing interest in the potential cognitive and longevity benefits of GLP-1 drugs, though definitive evidence remains pending. While some caution that the supraphysiological increases in GLP-1, thousand-fold above normal levels, could have unknown long-term consequences, the promising early studies highlight their role beyond glycemic control. Importantly, combining these drugs with exercise and strength training appears crucial for preserving muscle mass and cognitive function, enhancing their overall benefit. Snyder also mentioned emerging practices of microdosing compounded peptides to reduce side effects and cost, signaling a new frontier in personalized medicine. Snyder's lab has also dissected the broad category of dietary fiber, revealing its remarkable complexity and personalized effects on inflammation and metabolic health. Instead of viewing fiber as a singular entity, researchers highlighted various forms such as arabinoxolin and inulin, which differ in chemical structure, chain length, and physiological outcomes. In a controlled crossover study, these individual fibers produced divergent effects on cholesterol and inflammation markers based on the person's unique gut microbiome. Some individuals experience increased inflammation with particular fibers, whereas others show reductions, explaining why universal recommendations about fiber often yield mixed results. This underscores the interplay between dietary fiber and individual microbiome composition, established early in life but modifiable to some degree. Snyder's vision includes personalized fiber prescriptions based on microbiome profiles matched with probiotics to optimize gut health, reduce systemic inflammation, and support metabolic and immune function. This tailored approach could alleviate autoimmune and chronic inflammatory conditions that remain resistant to standard dietary advice. Throughout the dialogue, the critical influence of the gut microbiome emerged as a common thread connecting nutrition, metabolic health, inflammation, and even neurological outcomes. Approximately 70% of immune cells reside in the gut, where they interact extensively with microbes and dietary components, especially fiber. Snyder described how early life factors establish the personal microbiome guild that governs individual reactions to food and inflammation throughout life. Studies suggest that microbiome diversity has declined in developed nations as compared to indigenous populations, correlating with increased prevalence of obesity, diabetes, and autoimmune diseases. Lifestyle factors such as diet, antibiotic exposure, and environment intricately shape the microbial community, resulting in variable host responses. Integrating microbiome data with genomics and metabolomics could unlock personalized diet and supplement protocols to rebalance metabolism and immune function effectively. Moving beyond a uniform view of aging, Dr. Snyder introduced the concept of ageotypes, representing distinct metabolic and immune aging patterns apparent in different organ systems like heart, liver, kidney, and immune cells. Longitudinal profiling of individuals over more than a decade using multi-omic technologies, including metabolomics and proteomics, revealed that people age heterogeneously with variable organ system vulnerabilities and progression paths. This subtyping advances the idea of biological age from a single number, like methylation clocks, to an actionable, organ-specific assessment. For instance, individuals with markers showing a cardiac ageotype may benefit from targeted lifestyle and dietary modifications, as opposed to a one-size-fits-all recommendation based on whole-body biological age. Snyder described a commercial platform called Iolo that enables at-home blood sampling and metabolic profiling to help people understand and manage their personalized aging trajectories. Snyder's pioneering work in using a single drop of blood for deep multi-omic profiling has transformed the capacity to monitor health dynamically and non-invasively. Unlike the failures of previous ventures aiming at miniaturized blood analytics, his lab developed novel sample stabilization techniques that preserve proteins, metabolites, and lipids in small volumes suitable for mass spectrometry and RNA sequencing. Repeated hourly sampling over days allowed them to map intricate biochemical fluctuations in response to diet, stress, and exercise with unprecedented resolution. Such rich data sets enable correlations with wearable sensor information for contextual interpretations. As these technologies mature and become more affordable, they hold promise for democratizing health monitoring beyond the clinic, empowering individuals to track and optimize their physiology in real time. In complement to molecular assays, Dr. Snyder extensively uses wearable devices measuring heart rate variability, HRV, skin temperature, sleep stages, and galvanic skin response. He noted the growing accuracy of these sensors and the importance of avoiding overinterpretation or excessive daily focus, advocating instead for analyzing trends over days or weeks to reduce bias. These measurements provide key insights into stress, recovery, sleep quality, and autonomic nervous system balance. Beyond personal physiology, Snyder tracks environmental exposures continuously with portable air quality monitors that measure particulate matter, allergens, and chemical pollutants such as pesticides and carcinogens. By correlating external environmental data with internal biochemical and immune markers, his team aims to parse how air pollution and toxins influence inflammation, glucose control, and long-term health risks. Integrative monitoring of internal and external factors is poised to revolutionize preventive medicine. Venturing into mental health, Snyder's lab conducted innovative studies assessing physiological and molecular changes in participants attending intensive immersive events led by figures like Tony Robbins and Byron Katie. Combining wearable data, blood biomarker profiling, microbiome sequencing, and psychological questionnaires, they observed significant improvements in anxiety, depression, and burnout scores sustained for months following these programs. Though challenges remain, such as lack of randomized assignments, preliminary data suggest immersive behavioral interventions evoke measurable biochemical and neuroimmune shifts beyond placebo effects. This research helps bridge the often neglected connection between mental and physical health and highlights the importance of scalable psychological approaches that complement pharmacotherapy or traditional psychotherapy. Though often categorized as alternative medicine, acupuncture impressed Dr. Snyder for its reproducible physiological benefits, particularly in blood pressure modulation and inflammation control. Collaborations with experts studying mouse models have elucidated neural circuits by which specific needle placements regulate spleen function and systemic inflammatory pathways via the vagus nerve. In his personal experience, electroacupuncture sessions led to a significant and sustained decrease in blood pressure. This mechanistic insight validates thousands of years of empirical data and challenges dismissive attitudes toward acupuncture. It exemplifies how modern molecular and neurological tools can shed light on traditional practices, potentially expanding integrative approaches for disorders ranging from hypertension to diabetes and chronic pain. Finally, Snyder discussed how genetics contributes roughly 16% to lifespan variability, highlighting that lifestyle, environment, and epigenetic changes constitute the larger share of determinants. The longest human lifespans top out around 120 years, with centenarians often sharing traits like plant-rich diets, active lifestyles, and strong social networks. Notably, he recounted his own diabetes onset after a viral infection triggered epigenetic shifts, illustrating how genetics and environment interact dynamically over a lifetime. Advances in genome sequencing alongside deep phenotyping enable identification of at-risk individuals and personalized preventive strategies. Yet Snyder cautions that diseases are rarely driven by single gene mutations alone, but often emerge from complex interactions within thousands of genes and environmental inputs. Understanding this multi-layered biology is crucial for designing interventions aiming not only to prolong life, but also to enhance health span.